Thanks for coming for our kickoff for the core seminar series for the fall semester. Before I introduce our distinguished speaker, I just wanted to give you a brief overview of the Cell Culture Core Facility and what we offer. So the Cell Culture Core Facility is one of 30 here at UMass Amherst. If you go across the 5 campuses of UMass across the state, those add up to about 90 core facilities. It's not uncommon in cell culture, you need ways to analyze cells, so we have a very collaborative environment among the cores. Cell culture can interact with flow cytometry or light microscopy, as you can imagine, are quite powerful cores themselves. Primarily, we provide lab space, and that's in the form of biosafety cabinets, incubators, centrifuges, freezers, fridges, and we also stock consumables. Tissue culture uses quite a bit of it. So that can range from flasks to cell culture media, fetal bovine serum. So we have dedicated mammalian cell culture labs, and we also have a plant cell culture lab. We have a few researchers doing plant cell work, so they take advantage of those facilities well. We have a media prep station in there with water purifiers, analytical balances, and germinators for transferring cultures. So when I first joined UMass, my primary role was to preserve and continue this very large plant cell culture library that was donated to UMass from Monsanto. So there are over 2,500 species in the collection. Monsanto had developed protocols to cryopreserve some of them, but not all of them. So there are 1,000 species that are maintained in continuous culture. And as you can imagine, this is a huge undertaking to keep those alive, transferring them to fresh media all the time. So there's a double benefit here. In addition to providing this collection to any researchers in the world, we've shipped cultures to Tel Aviv, Israel, to Canada, and other points within the US. It also gives a great training experience for undergrad students who we hire to be in the facility and transferring these cultures onto fresh media. So some of the additional services that we offer in the core are just wet bench. You use my hands to get the work done. We can do anything. I list on the right-hand side here a few just standard services, whether that's cell line expansion and cryopreservation. We can produce stable cell clones by knocking in or knocking out genes. We do mycoplasma testing. We produce a lot of lentivirus for gene editing. And we can also do a lot of molecular biology, assay development, ELISAs, cell-based assays. I tried with one IELTS faculty PI, we have a basic training. So we'll just call that our cell culture boot camp, where we can take 4 students at a time and just teach them the basics of cell culture, aseptic technique, understanding the principles of the biosafety cabinet and how to work aseptically. Thawing cells into culture, maintaining that culture over time, passaging cells, and then freezing that culture back to cryopreserve a stock. So other than that, like I say, we're open to any other types of services and open to suggestions. So I'm going to introduce you now to our very distinguished speaker. James L. Sherley is the founder, president, and CEO of Massachusetts stem cell biotechnology company Asymmetrex. Asymmetrex develops and markets technologies for advancing stem cell medicine, including the first method achieving specific and accurate certification of the dosage of therapeutic tissue stem cells, called kinetic stem cell counting. Asymmetrex's kinetic stem cell counting platforms are also used to optimize manufacturing of therapeutic stem cells and for evaluations of drug effects on tissue stem cells. Dr. Sherley is a graduate of Harvard College with a BA degree in biology and the Johns Hopkins University School of Medicine, earning a joint MD and PhD degrees. Prior to founding Asymmetrex, he held academic research appointments at the Fox Chase Cancer Center, the Massachusetts Institute of Technology, and Boston Biomedical Research Institute. Dr. Sherley's professional awards include Pew Biomedical Research Scholar, Ellison Medical Foundation Senior Scholar in Aging Research, and NIH Director's Pioneer Award. So please help me welcome Dr. Sherley.
I tend to walk around a bit, so I think I'll put this in my pocket. Speaking of kinetic.
Exactly. So first, I'd just like to thank Mike for the invitation to come out and to share with you today. And I also want to thank him because he and the cell culture core facility here at UMass Amherst have been a really important partner in what you're going to see that we have developed at Asymmetrex over the past several years. And I just also want to just let you know that this talk has got plenty of time for questions as I'm going through, and there's time afterwards as well. So if there's something, a burning question, feel free to engage during the course of the talk. So there are a couple of important things I hope you'll take away from today's talk. And they're highlighted here. We're going to talk about this new technology called Kinetic Stem Cell counting, which now is available here at UMass Amherst, one of the first campuses where it's available. This is a technology for doing routine determination of the specific fraction of dosage or dosage of therapeutic tissue stem cells. And the most important thing to take away today is the importance of having specific counting of stem cells, why that's important, and also why it's important to be able to do it routinely, to be able to do it whenever you want to. So this is my conflict of interest disclosure statement. Everything I'm going to tell you about today right now is freely available from Asymmetrex here on this campus. But we are a for-profit company, and the things you're hearing about today, we're currently trying to hopefully develop as a commercial success. So this is the outline of the talk today. I want to start by just sort of reviewing tissue stem cells to get us all up to speed. Many of the people here, I think, are aware of these cells and their medical interests, but I want to make sure we're all on the same page, especially if we have students with us or anyone who's new to the field. Then I want to review why it's important to quantify tissue stem cells. It may be obvious to some of us, but I've been finding as we have been developing this company that it isn't apparent to everybody why it's important to even bother quantifying them. And then I want to introduce you to, if you're not already familiar with it, the stem cell counting problem. There is a problem counting stem cells. And I want to review the problems with existing technologies that are out there that have the goal of estimating the number of stem cells, the stem cell fraction of dosage. And then I'll discuss our solution, kinetic stem cell counting. And then finally, I'll just want to review how you can get access to kinetic stem cell counting here on this campus now. Okay, so let's just get started talking about tissue stem cells. This is a diagram here, the paradigm for how tissue stem cells renew and repair tissues in the invertebrate or mammalian body. So there are adult tissue stem cells. They undergo these asymmetric self-renewals that allow them to maintain the same stemless state while giving rise to cells that undergo differentiation. In many tissues, there's an initial group of cells that are committed progenitors that divide actively for a finite number of divisions before they give rise to differentiated cells. And depending on the tissue you're in, these could be differentiated blood cells, they could be hepatocytes, they could be intestinal columnar cells. Right? And that's a very important thing for us as human beings to be able to renew our tissues, because we grow up to fixed body size, but we continually make new tissue cells because of the activities of adult tissue stem cells. So I just want to go through a few examples of these tissue stem cell turnover units in tissues. So one of the best examples is the crypt villus system in the small intestines of mammals. And down here in the bottom of this crypt, there are tissue stem cells. They undergo asymmetric divisions, maintain their own state. They give rise to these transiently amplifying cells, which at the surface here, when these cells emerge at the surface, they undergo differentiation. They make 4 different types of columnar epithelial cells that do the function of the villus and the epithelium. And if you look out here at the very tip, roughly every day there's an apoptotic cell. So these are dynamic systems that are driven by the division of asymmetrically dividing tissue stem cells. Here's another example one of my old favorites, which is this is a section, a diagram section through a liver lobule. In this region called the portal triad, where there are these three vessels, the bile duct, a part of the hepatic artery, and hepatic vein, there's a place where their stem cells are not well defined, but they undergo asymmetric divisions to maintain their state. They give rise both to biliary epithelium and the hepatocytes. These hepatocytes migrate and differentiate toward the central vein here. And if you look here, at some frequency, you'll see a dead or dying hepatocyte. So again, the same process of stem cells dividing asymmetrically, giving rise to cells that undergo differentiation. And ultimately, you just stop dividing or die. And then probably one of the best examples of a tissue stem cell unit is the hematopoietic system. And this is a hierarchical diagram here of hematopoietic stem cells which undergo asymmetric divisions, giving rise to these early progenitors, which then delineate into lymphoid and myeloid progenitors, and ultimately give rise to all of these cells that form our blood system, many of which stop dividing and eventually die. So because of this role that tissue stem cells play in our human tissues, they are important in research to understand how tissues work. So there's a lot of interest in research with these cells and studying them in disease processes in tissues like cancer. And of course, in regenerative medicine, both stem and gene therapy medicine. So stem cell medicine is using stem cells, as we'll talk about, to repair tissues, to regenerate tissues. And then much of gene therapy is important with stem cells because the stem cells are the cells that are targeted for the gene changes because they have this long-term stability in tissues. So there are two main categories of stem cell medicine or stem gen medicine. A lot of it is with these renewing cells, so with things like liver or hematopoietic cells where the transplanted cells basically make more tissue. They make more tissue cells. They replace stem cells. But there's also a lot of work done in these stem cells like mesenchymal stem cells where it's the paracrine function of the cells that is thought to be important. So the secretion of cytokines and other factors that may either increase healing or reduce inflammation, reduce factors that cause pain. So, given these roles of tissue stem cells in research and in medicine, we can outline why they need to be quantified. And there are four main categories of the need for quantification. The first is in the process of doing tissue cell research. If you want to have quantitative stem cell research, you need to be quantifying stem cells. It's just that basic. Also, if you want to relate molecular properties and cellular properties to stem cells, you need to know the number of them to make those relationships. So for instance, if you're looking for biomarkers for stem cells in a sample, you need to know how many stem cells are in that sample to relate it to what you think might be a biomarker. There are a lot of companies that are producing stem cells and selling them commercially. So in the supply area, if you want to optimize tissue stem cells for biomanufacturing, you need to be able to quantify how many you have in your bioprocess. In the case of engineering for stem gen therapies, you need to know how many hematopoietic stem cells are present when you're doing modification of them. And after you've done modification, how many are still there? And in fact, there is an assay that companies use for this purpose, which I'll mention later. And probably the most important place where we need to know stem cell number and stem cell dosage for stem cell medicine. Currently, there are no stem cell medicines, so that would be bone marrow transplant, biological blood, cord blood, where the number of stem cells transferred to patients is known. And if you want to do the best medical development and medical treatment, what we know from pharmaceutical and biopharmaceutical treatment is that you need to know the dose of the critical agent. And right now, we don't have that in the case of stem cell medicine. Also, in stem cell clinical trials, if you are trying to relate the stem cells in your preparation to the effectiveness that you see, you need to have the right denominator, which is the number of stem cells that are in the treatment. And so having a way of quantifying stem cells could increase the statistical power of a lot of the clinical trials that are going on right now, based on stem cell containing preparations. And then finally, in pharma and biopharma, stem cells being important in tissues, they're important for drug action. Drugs that are activating for stem cells can be used for treating wound repair, for healing, increasing healing. And importantly, drugs that are toxic to stem cells, they will cause chronic organ failure. Because if you knock out the stem cell in an organ, basically it cannot reproduce itself. And there are many drugs that we know about that are stem cell toxic in, for instance, bone marrow and in the liver. So having a way to quantify stem cells will allow us to identify those drugs earlier. So our company spent a lot of time trying to say, we really need to be counting stem cells. If you want to see some specific examples of it, if you go to this website, for the past, I think, almost the past year, every other week we find a report in the literature, either a scientific report or a medical report that use stem cells in some way. We look at that report and show how, if they had quantified their stem cells, that research or that medicine would have been better. Okay. So, I hope what you get from this is that we think what we're doing in terms of figuring out a way to count stem cells is needed and important. And now we want to start heading toward what's the problem with having that information down. So there's something which I have learned over the last 8 years that not everyone, even people in the field, understand this about tissue stem cell containing preparations. And that is that they are not homogeneous for stem cells. So in this diagram that I'm going to be showing you, this is a flask. It's got tissue stem cells, and it's got committed progenitor cells. And the thing to appreciate is that in all cases, the stem cell fraction is small. If you think about it, it couldn't be otherwise. So there are many people who have the impression that their stem cell samples look like this, that they're homogeneous stem cells. They talk about them that way. They present them that way. And in fact, this could never be true because the starting sample from primary tissues, the stem cells are infrequent, they're rare, compared to the committed progenitors and the differentiated cells. When you make a primary sample, you start out with a low stem cell fraction. Now, there's been work to do things like enrich by selection and expand. But even in those enrichments, often this number here is still on the order of maybe 1% or so. So this never occurs. And since it never occurs, we're going to talk about why we need a way to be able to count these cells when they're not homogeneous. Okay? I also want to emphasize why it's important to know the specific number. The cures we're looking for are replacing tissues indefinitely in a person, long-term, durable cures. And to get that, you need long-term renewing tissue stem cells. These committed progenitor cells are short-lived. They can have some therapeutic effects, but they're not durable. And so for these long-term treatments, whether they're just the stem cells or they are gene-engineered stem cells, you need stem cells for that. And so the stem cell count is the critical determinant that we need to know, not the total cell count. Okay, so I always talk about stem cell specific dose. And I look forward to a time when you can say stem cell dose and everybody will be thinking specific. Right now, it's hard to know what people mean when they say stem cell dose. But in this case here, in this example, the stem cell specific dose is not 12, it's 2. That's the essence of what we're trying to do. Find a way to have a sample like this and be able to get that number and not just have this one. Now, the stem cell counting problem comes up because of 2 issues. We already talked about one. Generally speaking, stem cells are infrequent in the preparations that we have. They're rare. And that would be okay if we had a way of identifying them. divide them, to split them, to let them grow. And we do that until the culture stops dividing. It comes to a complete arrest. And with the data that we get at each passage, we generate what are called cumulative population doubling curves. You basically keep track of how many times the total number of cells double, and you just keep adding that up. And you get these curves that look like this. Some of you may recall that there used to be called Hayflick curves. For humans, every single human tissue you do will stop dividing at some point if you do the serial culture. Okay? That's our starting data for our analysis. Now, the way we get to the stem cell number that's responsible is we recognize that these curves, this data, cumulative population doubling versus time, has three features, three quantitative features. And what I mean by that, if you take different tissues from different people, from different kinds of tissues, and you do these curves, they will differ for the rate at which they go up, how fast they go up, that slope. They'll differ for the maximum number of doublings achieved, and they'll differ for when this arrest occurs. Okay? The factors that are responsible for those differences are two types of factors. There are what we call culture factors. And I can show you experimentally that these curves depend on the total number of cells I put in at time zero. If I put in different amounts, I get different curves. The split interval, how frequently I split the culture, determines what that curve looks like. The split fraction, how much I dilute the cells, affects that curve. And the viability of the cells. The cells are dying more rapidly or faster or slower. The curves vary, okay? We control all of these things. These are known factors. We either determine what they are in our study or we measure them. Now, there are these other factors that also this curve depends on that we don't know. So that curve depends on the numbers of all of these other cells, both at time zero and as they're being produced. All three of these cell types. Depends on their viability. We don't know what that is either. It depends on their cell cycle times. There are three of them. Cell cycle times of these guys, cell cycle time of this guy when it's dividing asymmetrically, cell cycle when it's dividing symmetrically. And then there are a couple of other things that are unique to this paradigm. This rate here, the tissue stem cell symmetric rate, how frequently a stem cell does this instead of doing that. And that's an important factor if you're trying to expand tissue stem cells, knowing what that is. And then there's something else that has to do with different tissues. The number of divisions that these cells undergo before they produce an arrested population varies. We call that the committed progenitor division number. How many divisions before you get a non-dividing lineage? Okay? There are two things we want to get. We want to get the stem cell number at time zero. That's what our initial goal was. We want to learn what is the stem cell number at the beginning of this, and we also want to know what the symmetric rate is. So when we do that, it's this cool thing called computational simulation. Okay? So we have this model for how cells continue to grow in culture, and we can use it to compute population doubling times. If we put in parameters for all these things that we don't know here, and the things that we do know, we can have this little engine generate data for cumulative population doublings. I'm showing you this schematically here. The dotted lines, that's the actual data. That is the experimental data set where we know these things. They're put into the model. And then these other curves here are what the computer simulates by randomly selecting these numbers here. There are about 10 of them. We just have it randomly sampled, put them in, turn the engine, get a simulation, and say how good is it with respect to the data. And what we're looking for is a selection of these numbers that gives us a simulation that looks like this, where it matches the data. And the only thing you'll have to be worried about when that happens, before you do some experimental testing of it, is whether or not you've got reciprocity going on. That's cases where these numbers right here this one in one simulation that looks like this. This number was low, and that number right there was high. You have another simulation. This number is now high, and that was low. We don't see that. All right, so what I want to tell you is that after, and I didn't mention this too, we do about 10,000 trials to look for the set that gives us the exact simulation. And what we learned is that this is a discrete set. There's only one set of values that we find when we get the simulation here. We don't find some other combination. They're quite discrete. So by taking a sample, doing a serial culture, getting the data set, knowing these factors from that serial culture, we can have the computer do simulation and find values that allow us to match that data set. And when we do, we get the stem cell number, and we get the stem cell symmetric rate. We also get all these other things as well. Okay? So let me take you through the process with a little bit of data. So this is a starting analysis. So we have a culture. I think these were CD34 plus umbilical cord blood cells. We're trying to count hematopoietic stem cells. So these squares here, or these, I guess these circles here, that's the actual cell count. So we're doing serial passage, we get a cell count. We do a serial passage, let them grow, we get another cell count. You see the cell count goes up and then it goes down. We convert that into cumulative population doubling data. That's that curve there. And then we do our computer simulation and we look for a set of factors that will allow us to simulate this data. This is an example of the factors that we get. And what we're doing here is we're taking the factors that we got and with some defined range around them, we're just randomly sampling and computing. And what you see is that there are 10 independent simulations here. And many of them you can't see because they're superimposed on one another. So we can do a really good job of simulating that data with the variables that we find. And once we get that set of initial factors, well, we're in computer space. We can compute the future of that culture. And that's what's shown here. So there are 3 types of cells that we get information about. Blue is the stem cells. And what you see for these cells here is well known. It's very difficult to expand these cells in culture. The stem cell fraction starts reasonably high, and then it just goes right down to near 0 as we do the serial culture. We see the committed progenitor cells start to appear, but then they are lost because they're turning into these non-dividing cells, which at the end of the culture, the simulation says there's nothing in the culture but non-dividing cells, and that's true experimentally. Okay? Right, so we can do this for any primary human tissue. So I need to talk a little bit about validation. Be happy to come back or talk with you one-on-one about the actual experiments, but we published this here in 2020 in OBM transplantation. There are 4 types of validations we've done. One, we have, because of our background in kinetic stem cell culture, we have these ways of detecting asymmetrically dividing cells in culture. So we compare those cytological numbers to what we get from the actual cell count. And they are within experimental error. So we can show that these asymmetric cell-renewal divisions are there, both cytologically and by what we count. We've done some analysis with agents that are known to be stem cell active or stem cell toxic. So you basically compare you count 2 samples, one with the agent, one without. There's a prediction when it's an activating agent, there should be more stem cells. There's a prediction when it's a toxic agent, there should be less. And we see good concordance when we do those analyses. This is a very important one for hematopoietic cells. That CD34 marker I told you about is expressed on both the stem cells and the committed progenitors, but not differentiated hematopoietic cells. So what you can do is you can take an anti-CD34 antibody, immunoselect out cells. So now you have 3 pots: starting cells, the positive cells, and the negative cells. And what we can show by counting those 3 samples, we get the expected result. The cells that are CD34 positive, we see an enrichment of our cell count. Our cell count is greater there. The negative fraction, we don't detect. At our level of sensitivity, we don't detect any hematopoietic stem cells there. So that's a really powerful one that we started with, and we have a new report coming out showing that using commercial samples. And then, as I mentioned earlier, we have compared directly now, head-to-head with a skin mouse for populated cell assay. We've done 2 independent trials. And to a first approximation, we get the same count that those skin mouse population studies showed. And that's in review right now with stem cells. Hopefully it'll be out soon. Reviewers willing. Just to, again, reiterate that we published this first validation studies in opium transplantation. It was the cover of that issue. And we were really excited because you see here, this is a cord blood unit, and normally there's no stem cell dose on it. There's a total cell dose, there's a CFU dose, there might even be a CD34 dose, but there's no stem cell dose. So we think the future is having a stem cell dose on these bags. Okay, I told you at the beginning that there was a 72-hour assay, and so far I've shown you things that take 3 to 4 weeks. How do we get to 72 hours? Math. All right, so once when I did this, I said, no, I should put this slide back in, but let's just go back. Once we do one of those 3 to 4 week studies, we get this data set here. Right? This is the stem cell fraction over time. No one's ever seen that before we were able to do this. So we get the stem cell fraction over serial culture time. And that means we can relate it to anything else happening on that time scale. So what we've learned from these analyses is that there are biologically determined equations that relate the stem cell fraction over time to the population development time of the culture. This is an example of one of them here. So if you have this equation here, at time 0 for this sample, if you just simply take it and culture it for 72 hours and put that population development time into this equation, it'll give you back the stem cell fraction for that sample. And if you take that sample and serially culture it, at any number of days afterwards, you put the number of days in, the 72-hour population development time, it'll give you back the stem cell fraction. This is a related equation. Once you know the stem cell fraction, you can take that sample and know what its stem cell fraction will be at some time in the future based on the number of doublings the culture goes through. Because this relates the stem cell fraction to the number of cumulative population doublings in that system. So that's what's available here now. We now have online calculators. I'll show you an example in a moment, where if you have a sample of cells, primary cells defined by these algorithms, you can culture it for 72 hours, any day in its history, enter that data into our online calculator, and it'll give you right back instantly the stem cell fraction. So here's an example of one of these portals. It may be difficult to see from back there, but basically, we have about 5 of these now. This is one for adipose-derived mesenchymal stem cells. So the algorithm is tissue source specific. Shouldn't be a surprise, that's important, it is. It also depends on what you cultured the cells in. So what this right here one is for commercial media from Thermo Fisher. It could have been some other media. We can figure out what that algorithm is and make it just for that media. There's some brief instructions about how to do that 72-hour culture. You enter the number of days of the history, the cell culture history, which in this case was 0. The time 0 count, the 72-hour count, the fact that you cultured them for 3 days, you hit that button right there, and you get the stem cell fraction. And I think right now, what's available here is, this calculator is available, and also the one for CD34 umbilical cord blood cells. I just want to give you some sense of the dynamic range of the method. So it has 3 orders of magnitude range. This is 4 different types of stem cell preparation. These are all commercial preparations. Here's CD34 bone marrow hematopoietic stem cells. That's about 2 cells per 10,000 total cells. This is adipose cells. Turns out those preparations actually have fairly high stem cell fractions. This is 64% stem cells. Right? So it's about 3 orders of magnitude dynamic range. This is one example of cord blood, which although there's less cord blood, so less total stem cells, the stem cell fractions of cord blood are known to be higher than in bone marrow. And so you see it's higher than what you see in bone marrow. But importantly here too, this is whole cord blood. This is CD34 fractionated. You see that significant increase in the stem cell fraction when we do that. So these are the stem cells that we've done so far to date. I highlight these ones in blue because they're now available at your cell culture core facility. So the method is, I won't say universal, but it's very versatile for different types of tissues, which is what's different than things like the skip mouse or populated cell assay. Right now, this is from a page on our website. We have 5 different online calculators available now. Most of them are for hematopoietic stem cells, you can see, and then there's this one here right now for mesenchymal stem cells. I want to be clear about this. These are the ones we did first with the resources that the company had available. We can make a calculator for any type of tissue stem cell source, primary source, in any culture media. I'm confident of that because we've done probably about 15 of these things now. These are just the ones that are best defined. So I just want to finish up with some acknowledgments. All the work I've told you about has been through funding that we received in the SBRR, the Small Business Innovative Phase 1 grant from the National Heart, Lung, Blood Institute. We're also a member company of this group called ARMI, the Advanced Regenerative Manufacturing Institute, BioFab USA. We received a technical project grant from them to do all of the automation of the software. Important collaborators because we just really need to work with other people so we can get outside of our own heads. And Mike Daley here at UMass Amherst, the core facility, has been really important in our development. And that's why these calculators are here first. Actually, the very first calculators are with Damien Heil's group, who leads the research and development group at ARMI. They actually have the first customized calculator for their specific cell populations. And then we have another academic group, Darnell Kegler at the University of Michigan School of Dentistry. A lot of work there with mesenchymal stem cells from oral tissues. And they have a set of calculators that they're currently evaluating as well. So these are some sites on our website for additional information. That's most of what I wanted to share with you today. I really thank you for your attention and your questions and comments so far, and I'm open and available for more. So.